Myself, Mr. Mate H.T., Department of Botany, RBNB College, Sri Rampur. So today we are going to study the T.Y.B.Sc Botany from this paper first BO 331, that is the cryptogamic botany. And from this paper, topic number 3, life cycle of the Batrachospermum. And in the life cycle of the Batrachospermum, we are going to study the reproduction. Reproduction. In Batrachospermum, there is two different types of the reproduction, that is sexual and asexual. Now there is one stage of this asexual reproduction which comes in the sexual reproduction. So we have to start from the sexual reproduction first, and then there is one stage in the life cycle of the Batrachospermum which is called as the asexual stage, that is in the Cantrantia stage. So we have to start from the sexual reproduction. So first, that is the sexual reproduction. It is advanced oogamous type. It is oogamous type because oogonia are dominant. That is, the female reproductive structure is dominant in the life cycle. So the type of a sexual reproduction is oogamous. Now here the plants are monoecious or dioecious. That means when both the male and female reproductive structure developed on one and the same individual, then such a type of a species are called as the monoecious species. When male and female reproductive structures are developed on two different individuals, then such a type of a species are called as the dioecious species. And both these species are found in the Batrachospermum. Here the male reproductive structure is called as antheridium, or it is also called as the Spermatia. And female reproductive structure that is the Carpogonium. Now both of these, that is the male reproductive structure which is called as the Antheridia or Spermatia, it is different, and female reproductive structure which is called as the Carpogonium is also different. So we have to first see the position and structure of all of these two, that is the Spermatia and Carpogonium. So first, that is the position and structure of the Antheridium or the Spermatia. These are the Spermatia, that is the spermatangium, and in the spermatangium there are the male reproductive structures called as the Spermatia. So this is the primary lateral. There develops a special branch. It is again rebranched. This is the spermatangium, and inside this there is the male reproductive structure called as the Spermatia. Now in Batrachospermum, on primary lateral, these are the primary laterals, and on the nodal region of this primary lateral, there develops a special type of a branch. So this is the special type of branch, and this branch is rebranched again. And the tip cell of each branch is get metamorphosed into a large circular cell. They are single or they are in group, and these cells are called as the spermatangia. At the initial stage of the development, they are green in color. So in each spermatangia, there is the male cell which is called as the spermatia. They have no locomotor structure. When they become mature, they breaks the wall of the spermatangia and sets free. They floats in the water. So this is all about the position and structure of the male reproductive structure, which is called as the spermatangium or the spermatia. They are developed on a special branch. These special branches are developed on the primary lateral, and these spermatangia are nothing but the circular structures. They are large as compared to the other cell. They are circular, and in each spermatangia there is a single spermatia. They have no locomotor structure. When they become mature, they breaks the wall of a spermatangia and sets free and float in the water. So next point. The position and structure of the female reproductive structure. So this is the trichogyne, upper part of the carpogonium. This is the egg. This is the trichogyne, and in between these two there is the notch. Here there is the receptive spot. This is the special branch called as the carpogonial branch. And this is the primary lateral. Now this is about the position and structure of the female reproductive structure, which is called as the carpogonium. Now in the Batrachospermum there are the two different types of the filaments, that is the main axis and the long branches. And on the nodal region of this main axis and on long branches, they develop the primary lateral. They are in group, and on this primary lateral there develops a special type of a branch. This branch is called as the carpogonial branch. It has only 3 to 5 cells. The uppermost cell is very large. It has a specific type of a shape, and it is called as the carpogonium. This is the female reproductive structure. Now this female reproductive structure is getting divided into the two parts. The upper narrow part is called as the trichogyne, in which just below the tip there is the receptive spot for attachment of the spermatia. Now toward the lower side there is a circular large part which is called as the egg, and in between this trichogyne and egg there is the notch. So this is complete structure of the trichogyne. Now this is the trichogyne. It is divided into the two parts, that is the trichogyne and egg, and this egg is haploid in nature. That means its nucleus is haploid. So this is about the position and structure of the carpogonium. Now next point, that is the fertilization. These are the enveloping filament. Primary lateral, egg, diploid, notch, trichogyne, this is the Spermatia. At fertilization, this is the situation in the carpogonium. Now, at the time of fertilization, very large number of the Spermatia freely floats in or on the water surface. By chance, they comes in contact with the trichogyne, which is the part of the carpogonium. Now, this wall of the trichogyne is mucilaginous, and because of this, very large number of the Spermatia are get attached to the trichogyne. But the one Spermatia which is at receptive spot is get attached to the wall of the trichogyne, and in between wall of these two get dissolved, and all the content which is in the male nucleus, that is in the male cell, that is called as the Spermatia, enters in the trichogyne. And from this trichogyne, the male content is migrated toward the egg, and then union of a male and female nucleus takes place, and here the fertilization is completed, and there is a formation of a zygote. So here there is a formation of a zygote, and when this content is get migrated toward the egg, then a mucilaginous plug is get developed here. So this is the mucilaginous plug to prevent the backflow of the male content. And when male content enters into the egg, then union of a male and female nucleus takes place, that is the zygote is get formed, and here fertilization takes place. And at the same time, the trichogyne starts to degenerate, and meanwhile these are the sterile filaments, they start to envelop this developing a zygote structure. So this is zygote which has a single diploid nucleus. Now this stage is at the time of fertilization, and this is at the time of after fertilization. That means the zygote is get formed here, and there is a degenerating trichogyne is there, and this is the mucilage plug. So there are number of changes which take place after fertilization also, and they are called as the post-fertilization changes. So we have to go toward the post-fertilization changes. So our next point, that is the post-fertilization changes. So all these are the which are seen in the figure. These are the post-fertilization changes. Before this there is the zygote, and zygote has a single diploid nucleus. First this diploid nucleus divides meiotically into 4 nuclei. That means these newly formed 4 nuclei are haploid. Later on these 4 nuclei divide again and again mitotically, and there is the formation of a very large number of the haploid nuclei in this zygote. Meanwhile, the wall of this zygote forms some outgrowths, and these outgrowths are called as the protuberances. So number of these protuberances are get formed. Now after this, one nuclei with some amount of cytoplasm get metamorphosed into this developing protuberance, and when it get migrated, then wall formation takes place and a new cell is get formed. That new cell is called as the gonimoblast initial cell. Meanwhile, these enveloping filaments are also encircles this developing structure. Now here, this gonimoblast initial cell divides again and again and forms a repeatedly branched structure. Now this is the branched structure, and all these branches are called as the gonimoblast filaments. Now each tip cell of the gonimoblast filament is get metamorphosed into a large circular cell, and that circular cell which stores large amount of the food material is called as the carbosporangium. So this is the carbosporangium, which is a newly formed structure, and inside this, that means in each carbosporangium there is a carbospore. Now as all of these nuclei are haploid, so this carbospore is also haploid. So in this way, very large number of the carbosporangium and carbospore are get formed, and meanwhile also very large number of this enveloping filaments are also get formed. Now combinedly, these gonimoblast filaments, these carbosporangium and enveloping filament combinedly called as a second stage in the life cycle of the Batrachospermum, and it is called as the carbosporophyte. So the first stage in the life cycle, that is the gametophyte, and second stage, that is called as the carbosporophyte. And in this carbosporophyte, especially there are the 3 different types of the structures. First, that is the gonimoblast filaments are there. Then there is the carbosporangium is there, and in the carbosporangium there is the carbospore and these enveloping filaments. So combinedly this forms a second stage which is called as the carpospore. Carposporophyte. Now when this carpospore becomes mature, they breaks the wall of the carpospore enzyme and sets free in the water. They freely floats on the water. After that, this settles on some solid object and there starts to germinate. And after germination, they give the third stage in the life cycle of the Batrachospermum, which is called as the Cantrantia stage. So we have to study the third stage, that is the germination of the carpospore and the development of the Cantrantia stage. So the next point, germination of the carpospore. Germination of carpospore. So this is the carpospore, which has a single diploid nucleus. This gives the germ tube. So next point, that is the germination of the carpospore and the development of the third stage, that is the Cantrantia stage. So this is the carpospore, which has a single haploid nucleus. Now at the time of germination, it forms a germ tube. So this is the germ tube, and this single haploid nucleus is get migrated into this germ tube, and this germ tube elongates into a filamentous structure, into a branched structure. Now at the initial stages of the development, this germ tube by division and redivision forms a number of branches and they creeps over the substratum. They are horizontal. So this type of a branch is called as the prostrate branch. That means all these branches, they creeps over the substratum. Now after this, these horizontal branches develop some erect branches. These branches are again rebranched, and these upright branches are called as the erect stage. Now in this stage there are the prostrate system is there and erect system is also there. Now this whole structure looks like another alga, that is the Cantransia. So this stage of life cycle in the Batrachospermum is called as the Cantransia stage. Now from the nodal region of this Cantransia stage there develops a new Batrachospermum plant, and here the sexual reproduction is completed. That means that is the development of the new Batrachospermum plant from the upright system of the Cantransia stage. Now next point, that is the asexual reproduction. Now here this is the Cantransia stage, and this upright system there are number of branches, and these branches are again rebranched, and the tip cell of these branches get metamorphosed into a large circular structure. That structure is called as the monosporangium, and in each monosporangium there is the monospore, and these monospore are also haploid, because this carpospore is a haploid, and this is only mitotic division are there, and finally there formed a new structure in the asexual reproduction which is called as the monosporangium, and in each monosporangium there is a single monospore. So this is also haploid in nature. That means this Cantransia stage is haploid in nature. Now these monosporangia or these monospore again germinate to give rise the same structure, that is the Cantransia stage. So this is called as the asexual reproduction in the life cycle of the Batrachospermum, that is the juvenile stage called as the Cantransia stage. So here sexual and asexual both the reproductions are completed. Now next point, that is the alternation of a generation. Next point, alternation of a generation. In the life cycle of the Batrachospermum, there are three haploid generations. Number one, that is the gametophyte. So this is the gametophyte, which gives the gametes. Second, carbosporophyte, which gives the haploid carbospores. These are haploid, this is also haploid. And third stage, that is the Cantransia. This is also haploid. So in the life cycle of the Batrachospermum, these are the three different types of the haploid generation, and only one stage, that is the zygote, which is a diploid in nature. So the type of a life cycle, that is alternation of a generation, is haplobiontic. It is called as the haplobiontic, or it is also called as the haplontic. So the alternation of a generation is haplobiontic, or it is also called as the haplontic. As there are the three generations, so it is also called as the triphasic, or it is also called as the trigenetic, because morphologically three haploid generations are there, that is the gametophyte, carbosporophyte and Cantransia. So this life cycle alternation of generation, it is also called as the triphasic or trigenetic. Thank you.